Hi, everybody. This is Steven, and Ben Hamstead asked me to do a short introduction to brain volumetrics. This is the sort of thing that you really could do an entire semester on, but we're going to try to keep this to under 20 minutes. And of course, I'd prefer to be there in person, but I'm down in Florida right now, and so we're going to do our best with this format. Speaking of which, I apologize in advance for any sound quality issues. We've just moved, and I'm recording this in basically an empty room with hardwood floors, so I'm really fighting the echo. If anything's unclear or you have any follow up questions, please feel free to email me at the address you see on this first slide, and just mention Hampstead somewhere in the title or the body of the mail so that it makes it into my priority queue. So by now you're probably already familiar with an MR image type called a 3D T1 anatomic or a 3D T1 structural image. Shown on the screen is a good example of a single individual's 3D T1 anatomic. It's really just a big rectangular box made up of many smaller rectangular boxes called voxels, and voxels just short for volumetric pixel. Those voxels, of course, are all identically sized, but the intensity of each voxel varies to represent the MR signal of the tissue that was captured within that voxel. We have very dark voxels, which have low intensities for cerebral spinal fluid, and very bright voxels, which have high intensities for white matter, and then sort of medium dark voxels in between representing gray matter. And obviously, those tissue type differences in intensity are what allow our visual system to find boundaries among adjacent structures. And if you look at the histogram of intensities from this image, you can see clear peaks associated with each of those three tissue types. You can look at that histogram and imagine how automated algorithms might use that distribution of intensities to try to find the boundaries between tissues, which is something that we're going to talk about in a little bit. By now, you're also probably familiar with the idea that a 3D anatomic image, like the T1 on the previous slide, can be turned into a map of the anatomic components that make up the brain in that image. Regardless of whether we're using manual measurement methods or fully automated measurement methods, the mechanics of how we measure volume are actually very simple. Each voxel in a structure of interest just gets assigned a unique label that identifies it as a member of that structure. So in this example hemisphere, many different structures have had their voxels labeled, and they're shown here with different colors because the display software I'm using associates a particular color with each label. We could just call this labeling brain parts, but because we like to sound fancy, you'll see this called structural segmentation or structural parcellation. And once that labeling has been performed for a structure of interest, it's very simple to calculate the volume for that structure. The computer just counts how many voxels were labeled as members of the structure and multiplies that voxel count by the known per voxel volume. So for example, the voxels in the cingulate gyrus have been labeled here and are shown in dark purple. If there are, say, 1,500 voxels that have been given the cingulate gyrus label, and the volume of each voxel is 1 cubic millimeter, then the measured volume of the cingulate gyrus is 1,500 cubic millimeters. So with that basic knowledge about structural images, I'm going to use the rest of my time to cover a few topics related to measuring volume. First, I'm going to give an overview of the techniques we can use to perform the actual voxel labeling. Second, I'm going to talk about how these approaches to measuring volume are different from something that you see in the literature called VBM, which is short for voxel-based morphometry. Third, I hope to convince you that this is worth talking about by introducing you to a few classic findings from the literature. And last, I'm going to talk about the methodological limitations of these kinds of volumetric measurements. These techniques are useful, but they're not without their problems. We can talk about the techniques used to label voxels in sort of three general classes: fully manual, semi-automated, and fully automated. I'll start by showing you an example of what it looks like to perform fully manual volumetrics. Here I'm using a program called ITK Snap to slice by slice begin to paint the caudate, the head of the caudate nucleus. And First, I'm using a zoom and pan tool to get the structure into position in all three planes in section. And now I'm going to switch over to one of these two tools to actually outline the structure. I'm going to start with the polygon tool, and I'm just clicking manually point by point to make a very careful outline of where I believe the boundaries between this structure and the surrounding tissue are. And once that's done, I click complete and accept, and you'll notice that all of the voxels get filled in with the label color that I'm using to label that structure, and that color has now appeared in the other two planes of orientation. As I complete a similar outline in the sagittal plane, I'll click complete and accept, and the voxels within that polygon will get filled in. And you can see in the coronal section, we can see both of those sections that I just outlined. And this will be my third slice, this on the coronal section. And if I connect to the starting dot and click accept, it also fills in. And if I come over to the 3D window and click update, I can now see a three-dimensional representation of that small portion of the head of the caudate nucleus that I've begun to manually segment. If I now change slices on my axial slice and switch over to the paintbrush and reduce the paintbrush to a reasonable size, I'm just clicking and dragging to fill in voxels with that label for the caudate nucleus rather than drawing a polygon and having the voxels within that polygon filled. Same effect, just a different tool. Sometimes the polygon is more efficient, sometimes the brush is more efficient. They both have the desired effect of assigning this particular label, which we've chosen for the head of the caudate nucleus, label 1, which happens to be colored in red, assigning that particular label to all of our voxels of interest. And I'm actually pretty fatigued during this measurement. This is about the speed that I would go at the end of a couple hours of measuring. In this case, I'm using a pointing device that I don't usually use for measurement. And you can see that after that second set of slices, we have something that even more closely approximates the head of the caudate nucleus. And if we were to continue going, we would fully segment the head of the caudate nucleus from the tissues around it, and by counting the number of voxels within it and then multiplying by the volume of each voxel, we would have the volume of the caudate nucleus. The identical output, which you'll recall is just a labeled set of voxels for a particular structure of interest, can be produced using a semi-automated method that might require less time and input from human operators. So on this slide, you see a time series of images showing an example of one semi-automated technique. In a software package called ITK Snap, the operator can place quote-unquote seed spheres within a structure of interest, and then the software's algorithms use the contrast within the image to try to find the boundaries of the structure. This can be much faster than fully manual labeling if the structure of interest has well-defined boundaries and the quality of the image is good, but it can also fail spectacularly with poor images or with structures that have difficult boundaries. In some ways, the computer has to work very hard to do what you do very easily with your visual system. In recent literature, Those manual and semi-automated voxel labeling techniques have mostly been replaced by fully automated whole brain labeling techniques like FreeSurfer. Whole brain labeling works by starting with a template brain, which you might also call an atlas. That atlas has pre-labeled voxels for the anatomical structures of interest. Software like FreeSurfer takes that atlas and its component anatomy and moves and rotates and stretches everything into alignment with your T1 image of interest. That's called nonlinear spatial registration. The result is that each pre-labeled structure from the atlas has been warped in space so that its borders now match the borders of the corresponding structure in your participant's T1. In other words, if you were to layer one image on top of the other image and make the top layer partially transparent, you would see that the structures and boundaries in the aligned atlas pretty much line up with the structures and boundaries in your participant's T1. And that effectively allows the anatomical labels already present in the atlas voxels to be more or less directly transferred to the voxels of your T1. That's a gross oversimplification, but basically how it works. This typically takes about 24 hours of computing time per brain, and it still requires a fair amount of human quality control and defect correction after those 24 hours of computing. So it's efficient, but it still requires some time and resources. One major advantage to using this method is that it labels voxels in what's arguably the brain's most complex structure of interest, the cortical gray matter ribbon. Its size and its convolutions and the number of regions it possesses with difficult gray matter cerebral spinal fluid boundaries make it really difficult for most labs to label it with manual or even semi-automated techniques. FreeSurfer labels the voxels in the cortical ribbon by first finding the boundaries between the cortical gray matter ribbon and the white matter that's immediately deep to it. Just think back to the intensity peaks in the T1 histogram that I showed you earlier and imagine how the software might use that distribution of intensities to find all the white matter voxels in the image and construct a surface that encompasses them. That white matter surface is shown here in yellow. FreeSurfer then inflates a copy of that white matter surface until it finds the boundary between gray matter and cerebral spinal fluid. That's the gray matter surface shown here in red. The voxels that are sandwiched between those two surfaces are then of course labeled as belonging to the gray matter ribbon. Not only is this a very efficient way to label gray matter ribbon voxels for calculating the volume of the gray matter ribbon, but it also provides a second metric, cortical thickness. If you take any point on that yellow-colored white matter surface, cortical thickness is just the distance between that point and the corresponding point on the red-colored gray matter surface. One of the things that's great about cortical thickness is that it's not just calculated as a summary value for an entire hemisphere or individual lobe or gyrus, the way that volume might be. If you look at those red and yellow surfaces and picture all of the individual spots where you can calculate the distance between the surfaces, it's easy to imagine how you could create a whole brain map of how cortical thickness varies across the brain surface. And in a few slides, I'll show you an example of how that kind of map can be used to analyze age-related cortical thinning. So that was a pretty brief overview of the range of methods we use to measure the volume of brain structures. Before I move on to some examples of important volumetric findings in the literature, I just want to point out that there's a difference between measuring volumes in the ways we just covered and an unrelated method called VBM or voxel-based morphometry, the output of which sometimes gets called volume, even though it isn't strictly volume in the way we just talked about volume. To perform gray matter VBM, each participant's 3D T1 structural image is first transformed into a blurred tissue type probability map. In that map, the intensity of each voxel represents the probability that the voxel contains 100% gray matter. That's an oversimplification, but a good way to conceptualize it for today. For all participants in a VBM study, these blurred gray matter maps are aligned in a common space. This means, for example, that if a voxel with coordinates ijk falls within the putamen of one participant, it's likely that voxel ijk falls within the putamen of all participants whose brains have been aligned for the study. This allows investigators to produce a whole brain voxel by voxel map as an answer to questions like, for each voxel in the brain, is there a significant difference between the gray matter of my target participants and the gray matter of my control participants? Or a question like, for each voxel in the brain, how strongly was my treatment group's gray matter signal related to treatment response? One example of a VBM study that you've probably heard of is the London taxi driver study. In that study, the number of years of taxi driving experience was shown to be correlated with gray matter VBM signal in the posterior hippocampus. VBM shows up a lot in the literature, and it's an important technique, so I just wanted to make sure that we differentiated between it and the sort of strict volumetric measurements we're discussing today. So now that we've covered the mechanics of labeling voxels and calculating volume and cortical thickness, let me try to convince you that this is all worth your time. In the next few slides, I've excerpted some interesting volumetric findings from the literature. This first finding is from a 2002 Nature Neuroscience paper from Harvard and the Boston hospitals. Not only did they find that smaller hippocampal volume predicts more severe PTSD in veterans exposed to combat, but it turns out that hippocampal volume reflects such strong genetic and developmental influences that the hippocampal volume of a veteran's twin also predicts that veteran's PTSD severity. This second finding is excerpted from a 1994 volumetric study in schizophrenia. The authors found that the caudate volumes in schizophrenia patients receiving neuroleptics increased in proportion to the dose of their neuroleptics. I like this study because it shows a dose-response effect, and it also links the anatomical effects of a pharmacotherapy to the pharmacotherapy's mechanisms, in this case, dopaminergic mechanisms. This third finding is excerpted from a 1996 Johns Hopkins study published in Brain. This is an early but important longitudinal study demonstrating that during maturation, the gray matter proportion of the brain decreases while the white matter proportion of the brain increases in volume. This final excerpt is from a 2004 cerebral cortex paper from WashU and Mass General. The authors found that cortical thickness decreases with age, and I just want to point out how much information about anatomical locations of the thinning can be represented in the figure just by virtue of the fact that the cortical thickness measurements can be mapped onto the surface of the brain. That map communicates a lot of rich information about the spatial distribution of their findings that would be really hard to communicate with tables or plots. That's a unique advantage to working with cortical thickness. So, to wrap up, let's talk a little bit about some of the limitations of these volumetric methods. The first limitation I want to talk about is just an inherent problem with structural MR acquisitions, and that's called partial volume averaging. If you look at voxels that are in the boundaries between two structures, really those voxels represent signal for more than one tissue type. So that makes it very hard to make a principled decision about where to place structural boundaries. And that's a problem for manual, semi-automated, and fully automated methods. There's really no escaping this problem, and we just have to do our best. The second methodological limitation I want to mention is also related to MR acquisition, and that's head movement. Even moderate amounts of gross head movement can introduce artifact and decrease the contrast among gray matter, white matter, and cerebral spinal fluid. And that makes discrimination of tissue boundaries difficult for the human visual system of any manual operators, and also for, of course, computer algorithms trying to use tissue contrast to find structural boundaries. Finally, one of the primary limitations of volumetrics is that they are time-consuming. If we're talking about manual volumetrics, those really require that you establish both within rater and between rater reliability, and so your n's are getting multiplied by the number of extra ratings that you're doing for that reliability. If we're talking about automated methods, they require manual quality checking and manual correction of flaws, and that's arguably something that you also might want to perform inter and intra-rater reliability on. The third point that I list here is that automated methods can require very precise skull removal. And actually, I want to extend that to all methods because frequently what you're going to be doing with your volumes from individual structures is expressing them as a percentage of total intracranial volume. And in order to do that, you have to have a very precise and reliable removal of the skull. That's how you're getting your total intracranial volume measurement. And so really that doesn't just apply to automated methods, but any method, automated, semi-automated, or fully manual, in which you're going to express your volumes as percentages of total intracranial volume. And then finally, I just want to point out that computationally intensive methods are computationally intensive. And that really costs both time and money. You can shorten some of the calendar time for those methods by running them in parallel, but unless you have a lot of spare hardware available, it costs money to run them in parallel. And so those computationally intensive methods do cost time and money, even though in some ways they're more efficient than a fully manual method. So that's all I have for this short introduction to volumetrics. I really hope that was a useful overview, and I'm also very sorry that I can't be there in person for your questions. Please feel free to email me if there's anything I can help with.